topic today, let me share the screens first. Antibacterial immune response. Can you see the slide, please?
Yes, sir.
Yes, sir. Yes, sir.
Okay, then. Okay, my topic is antibacterial immune response in Drosophila. I'm going to talk on the Drosophila as a model system, which has been used as a model to study the innate immunity, immune response against most particularly bacteria. We all know, I think by this time, I mean, everybody knows why Drosophila is a powerful model and why do we need to, we use this fly as a model everywhere. Like every branch of biology almost is using this fly model. Well, it serves as a powerful model for studying aspects of the innate immune system. The most important thing is to a maximum extent, around 75% of the genes shared by the human beings as well as fly. And most important discovery also that when the 2011 scientists got the Nobel Prize in Physiology or Medicine for the innate immunity discovery, which was discovered in the fly as a model. So it shares similar system compared to the human beings, especially innate immunity. It's a very complex, efficient, multifaceted processes, and it only possesses innate immunity, not adaptive immunity. We all know that there are two types of immune system like immunity that we come across, adaptive as well as innate. Innate is genetic and inborn. Adaptive immunity is the one which during lifetime, which an animal or organism shows to a particular specific antigen. Just a look at this slide. It says what are the different infections, including bacteria, fungi, viruses, parasites, parasitoids, all these things, similar to all organisms. Drosophila, being an insect, also has these threads from these microbes, which all infect this fly. As a result of this, fly shows many different kinds of responses by different modes, which include epithelial barriers, humoral responses, cellular response, and we also see the gut immunity, and we also see this involvement of the RNA phenomena in this innate immunity in the fly. Many other responses, including melanization, coagulation, different physiological reactions, including reactive oxygen species, and so on. An overview. Let me brief you how, in fact, this fly shows its immune system like activity of the immunity, immune response right from the larval stage. When you look at this third instar larva, we come across two types of immunity. Innate immunity is divided into two types, like humoral as well as cellular. Humoral immunity, we have the fat body, which is similar to this human beings' organ immune system, and it expresses antimicrobial peptides, which are similar to what is called as antibodies in human. And these antibodies, antimicrobial peptides, are very small peptides. They are produced by the two different pathways, like TOL pathway and IMD pathway. There are two different pathways involved in eliciting the immune response against the pathogens like bacteria. And we have what is called as lymph gland in the larva, which is responsible for the cellular immunity. which involves the different cells like plasmatocytes and crystal cells, lamellicytes, and each type of cell, kills the pathogens like plasmatocytes via phagocytosis. It kills them, and crystal cells are responsible for melanization. It responsible for the wound healing, and then wasp eggs like parasites, when they infect the lamellicytes, which encapsulate them and kill them. So this is just an overview of how Drosophila fly defends against pathogens.
Sorry to interrupt, sir. Can you keep it in slideshow view, sir?
Okay. Yes. Wonderful. That's. Can you see now? Is it okay?
No, sir.
When it goes to a slideshow, it's not visible, right? Can you see the slide now?
Slide we can see.
Now visible, sir.
I'm putting this into the slideshow. Is it not visible?
Click that.
Yes. Yeah.
But it is not coming.
Yeah. So I don't know how. Can anyone tell me how to put this screens? I mean here.
Sir, once double click there, sir, in slideshow, on slideshow symbol,
Okay.
2 times click, sir.
Slideshow symbol 2 times. You got it?
Sir, you twice you clicked, sir. No,
Yeah.
it's, no, sir.
You can press F5, sir.
F5. Can you see now?
No, sir, no.
One second.
But its PPT is clear, sir. No problem.
PPT is clear, and I'm just trying.
It is very small. That's why. This slide only is very small. Otherwise,
correct.
yes, sir.
ಸ್ಲೈಡ್ಸ್ ಹೋಗಿ ಸರ್ ಮೇಲ್ಗಡೆ ಟಾಪ್ ಅಲ್ಲಿ ಹಂಗೆ ಬನ್ನಿ, ಅಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಸ್ಲೈಡ್ ಶೋ ಅಂತ ಬರ್ತದೆ. ನೆಕ್ಸ್ಟ್ ಅಲ್ಲಿ ನೋಡಿರಿ. ಕೆಳಗಡೆ ಬನ್ನಿ. ಫ್ರಮ್ ದಿ ಕರೆಂಟ್ ಸ್ಲೈಡ್, ಸೆಕೆಂಡ್ ಒನ್. ಯಾ,
ಬಂತಾ?
ಅದ್ರ ಮೇಲೆ ಕ್ಲಿಕ್ ಮಾಡಿ ಸರ್. ಬರಲ್ವಾ?
ಅಲ್ಲ, ಕ್ಲಿಕ್ ಮಾಡಿದ್ದೀನಿ, ನನಗೆ ಡಿಸ್ಪ್ಲೇ ಆಗ್ತಾ ಇದೆ, ನನಗೆ ಸ್ಲೈಡ್ ಶೋ ಬರ್ತಾ ಇದೆ. ನಿಮಗೆ ಬರ್ತಾ ಇಲ್ವಾ ಸರ್?
ಓಕೆ ಸರ್, ನಿಮ್
ನಮ್ಗೆ
ಕಾಣಿಸ್ತಾ ಇಲ್ಲ ಸರ್.
ಶಿಫ್ಟ್ ಮತ್ತೆ F5 ಒತ್ತಿ ಸರ್, ಬರುತ್ತೆ ಅನ್ಸುತ್ತೆ.
ಓಕೆ.
ಶಿಫ್ಟ್ ಅಂಡ್ F5.
ಓಕೆ,
ಬರ್ತಲ್ಲ?
ನನಗೆ ಬಂತು, ನನಗೆ ಸ್ಲೈಡ್ ಶೋ ಬಂದಿದೆ.
Yes, sir. No problem, sir.
No problem, sir. Continue, sir.
One second, sir. Ready, sir. First day.
Or once you stop share, sir, again you share, sir. Do slide.
I'll do that. Yes, I'll do that.
Full slide na PDF ಮಾಡಿದ್ರೆ ಏನು ಚೇಂಜ್ ಆಗಲ್ಲ.
ಸ್ಟಾಪ್ ಶೇರಿಂಗ್ ಮಾಡ್ತಿದ್ದೀನಿ. ಸರ್, ಈಗ ಸ್ಲೈಡ್ ಸ್ಕ್ರೀನ್ ಬಂತಲ್ಲ?
Sir.
No, sir.
Sir, no problem.
One second, ನಾನು just ಇನ್ನೊಂದ್ಸ ಲಾಸ್ಟ್ ಚಾನ್ಸ್ ತಗೊಳ್ತೀನಿ. ಒಂದು ಸೆಕೆಂಡ್ ಟ್ರೈ ಮಾಡ್ತೀನಿ, ಸ್ಲೈಡ್ ಶೋ ಬರುತ್ತಾ ಅಂತ. ಬಂತ?
No, sir.
No, sir. You can move on, sir.
Okay.
You can proceed,
Okay.
sir.
I don't know how. It used to come. I don't know how it is. Use current resolution. Okay. What I can do is I can little zoom it.
Yes.
Okay, then I will do that now. Oh, I can't see this here.
Other contents will be not seen, sir, if you zoom it out.
Yeah. Okay. That I put less. The screen itself I will do maximize it. Okay. Is it okay now?
It's okay, sir.
Okay, sir.
Okay. 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 Let's begin with the components of the immune system of fly. It's exactly similar to the many components what we have. There are three different components: physical barriers, which include integuments and cuticle. It exactly corresponds to the skin and other integuments in our case. Cellular response, which include melanization, encapsulation, and phagocytosis. And humoral response, which includes the production of antimicrobial peptides. Okay. Physical barriers actually act as the first line of defense, which include the cuticle, gut, and trachea. Cuticle is a layer covering the external surface and the surfaces of foregut and hindgut, and acts as a support for the muscle attachment as well as functions as a protective barrier against environmental factors. Coming to trachea, is a branched tubular gas-filled organ. The tracheal lumen is covered by cuticular lining, which blocks the exit of the water, preventing dehydration. It blocks the entry of microbes and offering protection. So, yeah. And cellular response, which include the hemocytes. There are three types of hemocytes which correspond to blood cells: plasmatocytes, lamellocytes, and crystal cells. Each cell type has got its own role. Plasmatocytes, which account to more than 90 to 95% of the total hemocytes, present in all developmental stages of the fly, right from the embryonic stage to the adulthood that we see. And they are very small, round cells involved in phagocytosis. Phagocytosis is an evolutionarily well-conserved process of efficiently eliminating apoptotic cells and infectious agents by triggering a series of cellular cascades. Lamellocytes are a very small number. They are found only in the larval stage in very small numbers. They can be increased by infection with a parasitic wasp. When parasitic wasp infects the larva, then you will see the increasing the number of these lamellocytes has a response to this pathogen, and helps in the encapsulation of microbes and eggs of the wasps. It completely covers the wasp and form an encapsulation of these microbes and kill them. And then they're destroyed by reactive oxygen species, as reactive oxygen species is also a sort of response of the immune system of the fly, and which kill these pathogens. Crystal cells constitute the remaining 5% of hemocytes and are involved in the melanization. Melanization is the formation of deposition of the melanin at the site of infection or the injury, and that is synthesized and deposited at the site of injury, and it's similar to the clot, in the blood clot, or the site of wound what we see in our body. And that is it. Then at that site it traps the pathogens, and by synthesizing the prophenol oxidase crystals, cleaving of which form melanin and toxic intermediates. That is what you see at the site of injury or the infection. That was about the cellular response. The another response that the fly shows is the humoral response. Any pathogens attacks this fly, which this system gets activated by producing what is called as antimicrobial peptides. They are released primarily from the fat body and hemocytes into the hemolymph. And these basically AMPs, what we call antimicrobial peptides, are very small and cationic and are thought to affect the microbial membrane properties. And cell of barrier tissues also express AMPs as part of the local defense mechanisms. And these are what is the list. What you are seeing is there are the different parts of this larva, especially midgut produces diphtheria, atacin, and trachea produces drosophila and drosomycin, and oviduct also produces or secretes the drosophin and secropin, and seminal receptacle and the spermatheca also produce these AMPs, that is secropin, defensin, drosomycin. Yeah. So generally insect AMPs are synthesized as inactive precursor proteins or proteins. They are very active peptides, around 20 to 15 amino acids. They kill bacteria by membrane disruption. Membrane disruption means the membrane of the pathogen disruption, interfering with and targeting cytoplasmic components. The contact between bacteria and AMPs occurs through electrostatic hydrophobic interaction and strongly depends on the lipid composition of the bacteria. It depends upon the bacteria. As we know, there are different kinds of bacteria, including gram-positive and gram-negative and many other clinical strains. Yeah, I told you they are basically AMPs are produced as an endogenous peptides. They are as a very small, their size, molecular weight of 2 to 22 kilodaltons, produced by the fat body. It's an analog of the liver of the higher organisms, including humans. Interactions of these AMPs with gram-positive and gram-negative bacteria are different. This is another important thing. So meaning fly has not developed these AMPs against pathogens depending upon, for example, bacteria. Since there are bacteria which are some are gram-positive, some are gram-negative, depending upon the type of bacterial strain, these AMPs are going to be produced and interact. And total around 9 distinct classes of AMPs with the 23 members have been identified so far. As I told you, Drosophila lacks an antibody-mediated immune response that occurs in mammals, and it produces only antimicrobial peptides. Am I audible?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Just I wanted to make sure that. Thank you. So roughly 20 AMP genes, the antimicrobial peptide production produced by those genes. Around 20 AMP genes are in Drosophila fly have been discovered. Some are against antifungal, both against antifungal as well as antiviral and also antibacterial. And anti-gram negative AMPs produce atacin, secropins, and as I told in this case, as you can see here, the fat body produces these different AMPs, diphtheria, atacins. There are 4 members and 2 members of the diphtheria. Drosomycins are the 7 members, methicovin and defensin and so on. Yeah, this is just example here. Depending upon, I told you, depending upon the type of bacteria, a gram-positive bacteria, you see this peptide, specific peptide is produced, methicovin and defensin through tall signaling pathway, and gram-negative bacteria produce atacin, diphtheria, secropin, and drosophila through IMD pathway. Right. So, as I told you, this immune response by the fly is activated by different mechanisms. Mainly, there are 2 different signaling pathways: tall pathway as well as immune deficiency pathway. Tall pathway is activated by gram-positive bacteria, whereas IMD pathway is activated by gram-negative bacteria. The mode of infection is almost same, but the mechanism by which the immune response is shown by the fly has got its own mechanism, the route, and toll-like receptors pathway leads to the activation of this antimicrobial, some of the antimicrobial peptides in fly, and IMD pathway is activated by gram-negative bacteria. So again, it's a pathogen specific. Depending upon the pathogen, this fly has adopted this, as antibacterial, antimicrobial peptides genes activated. And mainly JAK-STAT pathway is not directly involved in the immune response pathway, but in only few cases like stress and injury, because it's a mainly pattern formation during which it is seen. That's not that we are going to talk about now. This signal-specific activation of AMPs is regulated by basically a nuclear factor kappa B particle and GATA-like transcription factors. As you know, transcription factors basically are triggered by these pathways. They are evolutionarily conserved and homologous to the mammals. We see this nuclear factor kappa B pathways, which are evolutionarily conserved, seen both in the fly as well as homologous to the mammals. For example, here, the toll, what receptor we have in the fly, we also have the homolog, similar gene, toll-like receptor or interleukin-1 receptor in human being. And we also have the immunodeficiency pathway, what we see in the fly, IMD, and the same mechanism is seen in the tumor necrosis factor receptor pathways in the higher organisms. The 3 NF-kappa factors in Drosophila are dorsal and different relish are the factors which are been identified. Okay, pathway gets activated during immune response of the infects this fly. Extracellular protease cascade lead to the cleavage, the prospagel to generate the ligand spagel, and you see this toll-like receptor, and with this MYD88, this is adapter protein which binds to the cytoplasmic region of this toll receptor and forms a complex. That is where it gets activated. As and when this gram-positive bacteria infects, and what you are seeing is receptor gets activated with the help of MYD88 adapter protein internally. Then you see this, pili which recruits another adapter, tube-like and tube kinase pili, and form MYD88 tube protein complex. And then what you are seeing is, it activates the different dorsal proteins. The factors which go on bind activate this DRS and mechincovin antimicrobial peptide genes. These are the genes which get activated by this gram-positive bacteria. And then downstream, when the complex is formed like this, it gets phosphorylated and degradation of inhibitor of NF-kappa cactus. And then you see this translocation of this factor to this complex called dorsal and dorsal related immunity factor diff into the nucleus. And then you see this, the activate transcription of various genes such as drosomycin, DRS, and defensin is another gene. IMD pathway is the immunodeficiency pathway, is initiated when peptidoglycan receptor proteins recognize mesodiaminomalic acid type of peptidoglycan found in most gram-negative bacteria. And that is why I told you the very specificity which makes this bacterial strain so specific and different, and the kind of the bacteria which can binds to this receptor to infects the fly, and you see this activation of their different pathways. So that leads to the formation of intracellular complex. That includes the IMD protein here, and then adapter protein, DFAD, and then space drain. So activation associated binding protein to complex dipterysin. So this is how this IMD pathway get activated. So yeah, finally this relish, the protein called 68 will translocate to the nucleus, and then it is a transcription factor which binds to this promoter of this gene dipterosin and activates this pathway. And this is one part that I tried to introduce to this background of the innate immunity, how immune response is generated or activated with the fly against the pathogens. The next part from this slide I'm going to present and share with you is a part of immune response or antibacterial immune response work, research work conducted in our lab. One of my PhD students, currently she is a postdoc in the NCBS Bangalore. She has studied and produced, a couple of papers from our lab now. So what we did was, we already knew some of the, how a fly shows the antibacterial immune response, what are the AMPs which are produced by the fly and other insects. We wanted to know, Drosophila melanogaster is a widely used species to study this innate immunity. We wanted to look into this, how a different Drosophila species with a different genetic background respond or elicit or exhibit this immune response to the different bacterial strains. To address this issue, we choose two different fly strains species. One is melanogaster and Ananaceae. They are genetically different species with a different genetic makeup, and they show a distinct immune response to any particular microbe. And we use these two different bacterial strains. One is gram-positive bacteria, that is Staphylococcus aureus, and another one is gram-negative bacteria, that is Escherichia coli. So we throughout this work, we use these two bacterial strains and how whether there is a difference in the immune response and genetically how this immune response or AMPs have evolved or adapted by the different species. Okay, here in this slide, we chosen this fly species. We chose those two bacterial strains. We infected these flies with these bacterial strains. We encountered this melanization at the site of pricking, and then we assessed some of the behavioral and survival studies and bacterial load count and so on, and fat quantification and nitrate production quantification. And the other side, we also counted like hemocyte, phenol oxidase assay, and antibacterial activity and so on in this world. And coming to these results of this experiment and setup, first we looked at this survival rate, whether both fly species have got the same survival rate or the ability to different bacterial strains. What we got is the rate of survival was higher in melanogaster species than Ananaceae. And that is because probably Drosophila melanogaster has shown more resistance to infection from both bacteria with the highest survival rate. No difference between bacterial strains. For both bacterial strains, melanogaster shows resistance. That means high survival rate. Whereas the rate of survival rate was varied after infection with S. aureus and E. coli. You can see this, even though both show survival rate, but the different rate, 65 to 49% and 49% to the S. aureus, and 74 and 59. 65 is for the South aureus, and 49 by the Ananaceae, 65% by melanogaster. For E. coli, melanogaster shows 74%, but for aureus, the Ananaceae shows 59%. Sorry, so that means there are two different species. So Ananaceae basically has the lowest survival rate than melanogaster. So this first thing is that we assessed whether both the bacteria, both the fly strains or species have got the same survival rate to the bacterial strains or not, that we assessed it. After that, we melanization, we as a part of immune response, it's a very clear indication that the site of injury or the pricking, you see the black spot here, what you are showing here, seeing here, a black spot, which is similar to the blood clot. It's found by these at the site of injury, infection. Melanin production is good indicator to microbial infection. It's shown, in both the species, and absolutely no difference here. And melanin production is little lower in Ananassia flies when compared to Malaviya flies. And basically, all these experimental setup were done at the different time points. Throughout the studies, we did this 3 hours, 6 hours, 12 hours, and 24 hours time points. Melanogaster shows response at the different time points, whereas Ananassia shows at different time points. So basically, it gives an indication. There is clearly a genetic bag which has a differential ability in this immune response to a particular pathogen. Just counted this, colony forming unit was higher in Ananassia than Melanogaster. So when there is a straight correlation, when there is a bacterial load was high in Ananassia, obviously it shows, especially at the 12 hours post-infection after SRES in both fly scenes, after which no significant CF was observed and they clear. Whereas in E. coli infected flies, CF is significantly increased till 12 hours to 24 hours post-infection Melanogaster. So bacterial load again is slightly different, and this was higher in Ananassia than Melanogaster species. Yeah, this is just a bacterial load and just plates. I can see this difference between all this SRES infection. What you are seeing is one side is a Melanogaster, the other side Ananassia, Melanogaster, Ananassia with the E. coli infection. Yeah, this is here, it's Ananassia species. Okay. So coming to hemocytes, cellular immune mediator is modulated during infection. What we have seen is there is a significant increase in the number of hemocytes in the SRES infection. And the highest level was found at the 12 hours post-infection, both the species. No difference in the hemocytes count for the SRES infection. Whereas E. coli infection, there is a significant difference in hemocytes was found in all the time points between Melanogaster and Ananassia. So what we are seeing here, increasing in the hemocytes is seen in 6 hours itself in the Melanogaster, whereas this increasing in the hemocytes number is seen in Ananassia species at 12 hours. So that means it's kind of like, it's slower and infectivity is slow, and it exhibits this response and only at 12 hours. And there is almost 3 hours difference, a 6 hours difference between the Melanogaster and Ananassia species. So fat quantification, we quantified the fat that deposition reduced the post-bacterial infection. What we got this between 2 bacterial strains, there is a significant reduction in the fat deposition at 48 hours post-infection in both the flies. And here, no difference that we observed in this case as far as fat quantification is concerned. As you can see here, almost a similar type of results are there, and then we moved on. So we also quantified nitrate level gets elevated during bacterial infection because nitric oxide is a very important factor as part of the immune response, is a very beginning of the immune response. It is expressed. And what we have seen here, SRES infected flies coming to this bacterial strain, SRES gram-positive bacteria, there is a significant increase in nitrate level till 24 hours post-infection Melanogaster, but in 12 hours only we can see this in Ananassia species. And coming to E. coli species, there is a gradual increase in nitrate level up to the 24 of both fly species. So the nitrate level significantly higher and for us in all groups except SRES infected, Drosophila Ananassia group in which nitrate level was reduced to 12 hour post-infection. So there is again 12 hours and 24 hours different is there as far as nitric level is concerned. Phenol oxidase activity is very important. That was the that is responsible for this melanization. That was increased during bacterial infection, and 6 hours in the melanogaster, phenol oxidase level significantly increased, but analyzer took 24 hours to produce this phenol oxidase, increasing this oxidase activity. So in E. coli flies, infected flies, an increased pH level was observed until 24 hours in both alpha flies. So you can see the difference only in different time parts, like only as far as SRS infection, but not E. coli. E. coli is a gram-negative bacteria. For gram-negative bacteria, both the fly species has got same ability. You can see the same phenol oxidase level activity, whereas SRES, a different bacterial strain, which is gram-positive bacteria, for which different species of the fly show different response. So again, it is an indication of that every species has got its own, innate immunity against a specific pathogen. Well, so this we have assessed in vitro antibacterial activity. Again, hemolymph from infected flies reduce the bacterial cell viability. The bacterial growth was significantly inhibited by hemolymph, because when we collected hemolymph and added to this culture and then looked at this bacterial load, and that reduced they inhibited the growth of the bacterial. So Drosophila homogenate inhibit the growth of the bacteria. What you are seeing is zone of inhibition. Yeah. So behaviorally, this was assessed by negative geotaxis. Is one of the important factor or the experimental assay that we do, either stress physiology or immunological aspects, whatever. Fly negative geotaxis is one of the parameters which is to be studied, behavioral assays. So there is a variation negative geotaxis movement. Basically, the fly moves upward, and when the vial or the bottle, when you culture the flies, and the flies start climbing or the moving upwards, and this has been counted in a number in a specific time, in 1 minute or 2 minutes, like whatever the time duration, how long the flight takes, whether it shows the negative geotaxis or not. So infection, bacterial infection cause changes in this locomotor behavior. Definitely we got these results, like rated in post SRS infection, a significant decrease in the climbing behavior, whereas in E. coli infection, we could see this after 12 hours, this negative geotaxis. So coming to this basic immune response, all this I explained you, these results I gave you are the basic immune responses shown by the fly, different species, including survival rate and phenol oxidase activity, melanization, and zone of inhibition and immunocompetency. The results of this, the experimental setup have shown that if there is any immunocompetence of the laboratory stocks of the melanogaster and analysis species against these two bacterial species. So in some of the aspects, there is a difference. In some of the aspects, there is no difference. Especially E. coli, there is no much difference, but SRES is for the gram-positive bacteria, they could see this species-specific specificity in immune response. And after this, we wanted to characterize these AMPs in both the species. After this, again, we infected the fly, the bacterial infection, and then we extracted the hemolymph by the tissue homogenization. We did HPLC profiling, and then SDH page or LCMS, and then we also analyzed this phylogenetic analysis of the AMPs. Okay, so what the results what we got? Basically, we quantified the protein concentration at different time points at the 24 hours. For example, melanogaster, produced 69.4 is the amount of the protein concentration against SRES, whereas NNSA is 114. So the higher level of the protein produced, a total protein quantified against this bacterial infection. Just we wanted to know whether there is a difference in the protein concentration or not. Again, E. coli, there is 82.1 and 104 in NNSA. So NNSA species has produced highest level of the protein against these two bacterial infection, saying that there is a variation in the protein concentration between fly species after bacterial infections. So meaning there must be a different set of proteins. After this, we moved on to analyze the proteins, what exactly the specific proteins expressed in different species. We come across what is called as differential expression of immune-induced molecules, AMPs, among Drosophila species. And what you are seeing is this HPLC chromatogram. This is the E. coli and SRAS and control. This is a melanogaster species and NNSA species, what you are seeing here. And the spectrum is different, from which we selected specific protein components and analyzed by SDS page electrophoresis of the hemolymph and densitometers. And there are several proteins are up and down regulated during infection of these two species, these bacterial strains. And melanogaster, we could see this 58 and 34 and 130 kilodaltons, 3 different molecular size protein identified. And in NNSA, 45, 33, 27, 14, and 11 kilodaltons, which are specific to this NNSA, we could analyze it. Then we moved on to analyze trypsin, as compared to this previous slide. We went for trypsin SDS page hemolymph because it could give us a still clear expression pattern of these proteins. It's called as was detected in E. coli infected flies. A single protein band of around 6 kilodaltons small was differentially produced in melanogaster and NNSA infected, but not with the SRAS. So there is a difference here as far as this protein is concerned. Both the species show differential expression against E. coli, but not SRAS. So this is the E. coli, what you are seeing here. Let me zoom it a little bit as this. Yeah, so there is a slightly specific differential expression of this 6 kilodalton protein here in a different species, only in E. coli infected, but not SRAS. Then we went for amino acid sequence and fragmentation of spectra for tryptic peptide by LCMS spectra. We could identify these amino acid sequence which are of these 6 kilodaltons. We could see this, and in the database we could blast it, and then we could identify this molecule, what is called a secropin A, in which red colored sequence represents the tryptic peptide, and its spectra sourced from the hemolymph and melanogaster NNSA species. Yeah, so based on this Muscat search, we could identify this AMP, the peptide was putative secropin A-like peptide, and this is the amino acid sequence in both the species. LCMS detected sequence is this is the amino acid sequence, and the molecular weight is this, and the score that got into Muscat analysis is such 111 for the melanogaster and 58, and protein identified in secropin A, similar to the Hyloporus cecropia. This is which is a kind of protein which is already identified in different insects, which has given the accession number in the UniProt KB database. And this is a Dalton's is a mass of this protein, and sequence coverage was around 26%, not much of it. So that means this must be this secropin protein like peptide might be a little more different than which is already identified. The mass spectrometry proteomics data have been identified. This Proteome Exchange Consortium OYA Private Partner repository. And this we have reposited in the databases, and each accession number with this DY given to this Malaviya analysis species. And when we compare the sequence of all the secropins available in the database compared to our sequence, and you can see and identify the, dendrogram, that what we can see here. Secropin A, A1, A2, B, C, and C2, and there are similarities, and based on the similarities, what we are seeing is the closely relatedness of these two different. And you see here, Secropin A is Ailoporus secropis as taken for as a reference group, and Melanogaster Secropin A like here, and similarly Drosophila analysis. What you are seeing is this, even though in some of the basic human responses that we could see the difference, but this AMP around 6 kilodalton protein that we have characterized here share the almost same homology, and NNSA and Melanogaster share the same ancestry. So this has been diverged. Drosophila analysis secropin A1C and 2, they are diverged from this common ancestry, and you also see the secropin A1 and A2 here. And they all have this, again, common ancestry from secropin A1 to Drosophila melanogaster, and secropin P as the common, ancestor for all these things. So, sequence alignment, the amino acid sequence, what we have identified from LSMS analysis, background correlative with other secropins. And what we got here, in E. coli infected flies, 6 kilodalton band was produced higher levels. LCMS data and mascot analysis has identified this putative secropin A-like peptide, and that suggests that both species of Drosophila have exhibited clear immune response, no doubt. The flies were also able to discriminate between bacteria, as putative secropin peptide was produced in flies infected with E. coli, but not SREs. So, secropin A-like peptide that we could see only against E. coli bacteria, but not Saccharomyces, Staphylococcus aureus strain. So this is a kind of, this is a end point of this identification of this peptide. It still further needs to be investigated. By the time, we could ended up, and from here onwards, we all continued identifying this immune challenge. The flies were already exposed or infected with this against bacterial strains, also studied for the immune priming or challenge. That I'm not going to present here, and even that also has proved as give us the evidence that only against a bacteria that is gram-negative bacteria E. coli, against this secropin A-like peptide was produced, but not SREs. So, yeah, let me conclude these with these results. The differences in the physiology of the diverse species against specific bacteria and variation in the species resistance, and also heterogeneity in flies. And conclusion point is analysis species show high sensitivity to bacterial to the infection, and this reflects significant variation in the species resistance. In addition to this general melanogaster innate immunity or immune response, we wanted to check this any differential specificity or immune response variation in different genetic background, that is melanogaster and NNSA, and this is what we could end up with this now. So I think